Thank you very much, Professor Kim, for the overly kind introduction. And first of all, I would really like to congratulate Sherry, David, Leslie, and the rest of the IOC for putting together such a wonderful meeting. Thanks for bringing the ISF back to the Asia-Pacific region. This is actually the first ISF annual meeting after Kyoto back in 2019. And I still remember actually the first time when I still served on the board as a member at large that Melbourne was first proposed as a candidate city for the ISF. I just feel like thrilled to see this actually come to true. This is wonderful, really exceptional. So my name is Hubert. I work at this university called Tsinghua, based in Beijing, China. And we try to set up the link between basic research and applications. So I would like to have a disclosure statement also. So I'm also a co-founder and a shareholder of Carrier Biomed, which is also a round sponsor of this ISF 2024. So in the, I guess, the frontier spirit of Australia, I would like to show you some of this newest development of our lab. So if some of this, actually a substantial portion of the talk today was first disclosed, disclosed for the first time, so if some part is not really polished, I apologized beforehand. So the focus of our lab is innate immunity, right? Especially after the 3 years COVID, we do know vaccine and all of the other immunity response. So I still remember at the beginning of last year, I hosted Julius Hoffman from France at Tsinghua. So actually, even after working in this field for close to 20 years, I still actually learn new things. Dr. Hoffman won Nobel Prize back in 2011 for the discovery of toll-like receptors. And he talked about toll-like receptor, of course, is a major part for our innate immunity. We were born with this self-protection. And in the immunological response, innate immunity could count for over 70 or 80%, so the majority. For you guys who speak German, you'll know toll in German means amazing, right? So these receptors, indeed amazing receptors. They're just like those guards on the watchtowers of the Great Wall, like in northern China. They would actually detect the pathogen or danger-associated molecular patterns and turn on self-protection. For example, the very first ligand for toll-like receptor is LPS. We have already heard a lot of stories associated with LPS in the last 2 days. So LPS is a component of the bacterial cell wall. And for people like you and me, we don't have cell wall on our cells. So when we detect LPS, we would know there's some alien invasion, if you will, and we turn on our self-protection. So apparently, if you have too little of this protection, you can't protect yourself. If you have excessive protection, you'd have autoimmune disorders. So to precisely control the innate immune response is the key. And since I started as an independent PI at the University of Colorado Boulder back in 2007, we have been working on just that, to develop small molecule probe for toll-like receptors. So our group reported the first small molecule receptors for toll-like receptor 1, 2, 5, 3, and 8. We also work on some of the agonists also, because those toll-like receptors, they are double-edged sword. You just want to control these. And in the last, I would say, 4 or 5 years, we focused on this endosomal toll-like receptors, such as the 7 and 8, because there was no small molecule inhibitors for those crucial innate immune receptors before. So, for example, toll-like receptor 8 detect single-strand RNA, especially virus associated single-strand RNA, just like SARS-CoV-2 that caused COVID. So we do know that excessive toll-like receptor 8 activation was associated with autoimmune disease such as rheumatoid arthritis. So we reported the very first toll-like receptor small molecule inhibitors, and in collaboration with a hospital based in Beijing called Peking Union Medical College Hospital, we showed that could be a novel way to, as a candidate drug, to actually attack this autoimmune disease. Because even like we have like biologics, there's still like not very efficient and safe small molecule drugs for those autoimmune disease. We also tried some rare disease such as adult onset Still's disease. Even now, so I still got emails sometimes saying, Dr. Yin, I saw your paper in this Nature Chemical Biology. My sister got adult onset Still's disease. Can we get on your clinical trials? So there's definitely like still much need for in that area. So you know, as a basic scientist working in the lab, I sometimes underestimate the efforts to push things from bench to bedside. The paper was published back in 2018. What did we do in the last 5-6 years? We actually we work with like different teams for the safety evaluation, for the production, for all of the pre-IND. Finally, last year, 2023, this first-in-class candidate drug was approved by US FDA as well as the Chinese CDE for clinical trials. So we just actually finished the first batch single ascending doses and shows pretty good safety. And right now we're actually like starting the multi-ascending doses for the evaluation of these new drugs. So for these toll-like receptors, what we talk about, we humans have 10 different toll-like receptors, just like TLR4. But EV apparently also play an important role in this cell-cell communication in the innate immunity response. Besides the surface receptors, such as TLR4, LPS is known also could be wrapped up by outer membrane vesicles and get into the cells, turn on program cell death, such as pyroptosis. And a co-PI in my lab, Dr. Ying Zhang, also asked the question, did EV, like many other things, like, you know, we know extracellular vesicles were like really perfect, really good at doing, also mediate the innate immune cell-cell communication. So this was published work. I'll just like sum up really quickly. Ying showed that TR9, which was activated by ODN DNA, which is a specifically methylated DNA molecule from the bacteria, could actually activate the cells. And in the EV secreted by these activated cells, they would actually like carry out those cargoes. And upon uptake by the recipient cells, they would turn on the inflammatory consequence. And also by remodeling the cytoskeleton, actually like have a feedback to regulate this important process. So this actually taught us maybe for this crucial, important innate immune response, EVs would play a crucial role in this cell-cell communications as well. And more recently, we also asked for bacterial EVs function in this cancer microenvironment modulation. We have already heard like very exciting stories from many of our colleagues, including Yang Song, for example, on the bacterial EVs roles. Apparently, those bacteria live in the guts, have many different impact in our health. And Dr. Jingyu Li in my lab worked with the Beijing Cancer Hospital, showed the bacterial vesicles facilitate actually colon cancer metastasis, especially the clinical relevant colon cancer liver metastasis. She also showed that the bacterial EV could actually have some efficacy to actually like suppress the immunological response, and also actually like may also actually suggest some like novel therapeutic strategy also. So here I want to show this movie because I guess a lot of you guys already see a different version of this. I just stole this from CCTV, which is a network in China. And this actually was actually a short movie prepared by NHK that Yan Lat-Bell, Takahiro Ochiya Sensang actually helped to prepare that. So apparently, actually this EV already penetrated to almost every single aspect to our biomedical research. In China, we found CSF back in 2017, and last year, I know, we know that guy, right? So last year already actually in the medicine division, actually over 40% of the proposals have some EV component. This actually is really quite impressive. Thinking about this is still like relatively young field because EV can do so many things. So a little bit like history. Again, as I mentioned, I started my lab back in 2007 in Boulder. And we started actually a Howard Hughes CIA project. That stands for Collaborative Innovation Award. But actually the abbreviation sounds interesting, CIA. But basically we just try to develop a curvature sensor to actually sense the tension of the membrane surface and come up with a peptide or other probe for sense that. So a postdoc in my lab, Jonelle Salud, along with the graduate student Leslie Morton, started our efforts in EV. This was back in 2009. And Jonelle, after that, my lab and Jonelle's lab, after finishing postdoc in my lab, he became assistant professor at the Washington State University and then moved back to his home country, Philippines. So this actually like Jonelle's work in Colorado. Actually, this is a painting by another student in my lab, and later on get the cover of a journal. This tennis ball was what fluorescently labeled peptides that specifically target EV. And actually, it's interesting. So I went to visit Jonelle in March this year. Right now, he's the vice president of University of Augustine at Iloilo, Philippines. This actually is a remote island, actually like 2 hours flight from Manila. But I felt pretty touched because I saw even his office actually it's almost like a reproduction of our office back in Colorado. And he set up a research center called C2B2 at this remote island in Philippines with a major focus on EV research. Right, you guys know that Philippines has a lot of really interesting natural product from honey, from pineapple and things. So I just feel like this is where Iloilo is. This region of Philippines. I was thinking really our efforts 15 years back in the States started to bear fruits, even touching a remote corner of Philippines. So. And then, as Dr. Kim mentioned, I'm in Tsinghua. So I moved from Colorado to Tsinghua full-time back in 2018. And again, EV remained a focus of my lab. So this is some more recent work. A student, Dr. Xue Jin, actually a student from our arch rivalry, Peking University, reported LPS could actually stimulate, right? LPS actually is a TL4 ligand, could stimulate cells, and we saw each of this blinking is a secretion events of EV secretion. So she was asking the question, maybe for innate immunity, right? We know this is important, but too much immune response is no good either. So this could be a way to control excessive immune response. So when Xue looked into this, he found an interesting molecule, PI4P, which is a phosphorylated version of PI, this lipid, play an important role here. We know PI well. PI is the second messenger that regulates many important biological processes. So we're asking, really, what's maybe the lipids also play a role here in this LPS-induced EV secretion. Talking about these, we sometimes, like yesterday, we heard the leader of the field talk about all of the omics technologies. There's so many different omics, genomics, proteomics, but really talking about lipids, I feel that we're still lagging behind. This is more like we're tell a war story. It's easy to focus on those generals or knights, but really like the people who went to the battlefield and fight as those soldiers. So lipids are indeed those soldiers in our war against different things. So we humans have more than 1,000 different lipids, and their abundance, their distribution, for example, PS, this negatively charged lipid molecules in normal cells, 99% of the PS pointing inward. Only when cells undergo apoptosis, for example, the PS got flipped out, and our immune system recognizes them and clears them out. This is so-called eat-me signal, as many of us know. But coming to the lipids in EV, it's still hard to study. One of the reasons, unlike proteins, you can use antibody and other probing technique. For lipids, oftentimes you really have to look into their intrinsic characteristic, which is their molecular weight. So we work with chemists at Tsinghua, the Xia Yu's lab, trying to actually develop new chemical biology tools to study the lipid. For example, we know where the double bond locates on the tail of the lipids matters because they actually really affect the shape of the lipids, and also the cis and trans isomers, they would have exactly the same molecular weight, but their function would be very different. So we actually use chemical biology method to form the four-member ring by different cleavage pattern. We can tell where the double bond is and what the cis-trans isomer is. We also actually use fingerprinting technique to study different phosphorylated location, really different regional isomer, if you will, that's a chemistry term, and study their different lipids function. So for these, PI could get phosphorylated once, not only once, but only with a dual phosphorylation also. And each of these phosphorylation or dephosphorylation step would be controlled by different enzyme also. This is a very dynamic, complex system. So actually, this is indeed actually hard to study. To make a long story short, we actually combined bioinformatics and biochemical method and showing that this PIP5K1C to phosphorylate PI4P to PI45P2 actually is the key for LPS-induced EV secretion here. And because this was already published last year, I would make this a long, summarize the story. We think LPS could actually activate TL4 and turn on the interferon pathway. And by recruit PIP5K1C, would recruit this thing into the endosomal sorting and involve heat shock protein and the RAP family, which actually was also known to play an important role in autophagy and other things. So that's maybe one of the ways to control excessive immunological response that could actually facilitate EV secretion. So this actually may give us some clue how this excessive innate immune response was actually regulated by EV. Remember, in the old days, we do actually think EV could be the trash bag actually you throw out after dinner as one of the ways to regulate the immunological response. We talk about toll-like receptors so far, and toll-like receptor already won a Nobel Prize back in 2011. So what's beyond these amazing receptors? In my personal opinion, I think SIG-STING pathway is really quite important. I still remember back in 2014, I invited my good friend, Professor James Chen from Southwestern Medical Center to Boulder to give a talk. The very first slide James actually presented was that immunity beyond toll-like receptors. So he was talking about this back then newly found innate immunological sensor in the cytosol. So unlike those toll-like receptors we found in the endosome, right? So far we talked about toll-like receptor 8, for example. They were found in the endosome. They're ectodomain pointing to the lumen of the endosome. But the C-gas, since double-stranded DNA, which actually usually is not actually like commonly found in the cytosol, and turn on the synthesis of C-gap, and then turn on the STING pathway. So far, we do know this could be a very essential pathway response, especially in the innate immune system. So we work with James' group on development of like novel therapeutic strategy to regulate C-gas STING as well. But so far, we haven't actually put anything into the clinical stage yet. So we think actually there's still like a lot of new things to study, even like this already like, the C-gas was firstly discovered back in 2013, over 10 years ago. There's still a lot of new things. For example, we reported March 8 could be a non-canonical path to regulate this important group of innate immune receptors also. March 8, we reported last year, March 8 is the new E3 ligase regulating C-gas by putting the ubiquitination on the K63 sites, and it could actually like really like inhibit the double-stranded DNA binding to C-gas. And we think this could be a way to a new way to get around the current failure in the drug development for the C-gas STING as potential autoimmune disorders regulation. So this paper was already published in Science Cycling and actually was reported by the Science Journal also. So it just actually shows how much we still need to learn in this important pathway, and this actually could actually have a potential in technology translation also. STING is the same. STING is downstream to C-gas. Last year, to that LPS paper, we published back-to-back in the Journal of Extracellular Vesicle about STING's excessive immunological response regulation. This was done by student Jiaqi Liang in my lab. So Jiaqi actually showed upon STING activation, both in the cell and in the EV, STING and phosphorylated STING's level got significantly elevated. So he found this actually is interesting because the STING actually like co-localize also with the multivascular body marker VPS16. So we were asking we do know actually STING undergo a lot of like many different layers of regulation, right? STING actually upon dimerization and further tetramerization could translocate between Golgi and ER. And we actually found a protein called STEM, which is a part of ASCORT0. Actually, this again is interesting. Regulates the secretion of STING from those ER actually get into the in the vesicle and then eventually got secreted to the external of the cell. So we found actually upon the STING, the oligomer, again, let's talk about the oligomerization regulates the translocation of STING. STEM is induced. The expression level was elevated, and then it basically pushed the STING out. So maybe, again, we talk about this is important, but it's also very general. So cells got to have multi-layers of control of these. So just to avoid excessive innate immune response, maybe this is a way for STEM, actually it's a new mechanism, machinery to control excessive STING activation. However, as we just talked about, STEM is a part of the ASCORT0 pathway, and I'm sure many of you guys would be very familiar with this secretion machinery. We found STING, actually this function for STEM's regulation of STING is independent to the conventional path, conventional function of the ASCORT0. So when we first saw this, to be honest, we feel we're pretty hesitated. Is this true, or is this just an artifact? And then luckily, after our paper was published in March last year, we saw a couple of new papers also reporting this new function of STEM. So I guess actually a lesson we learned from this is that it seems actually like, a lot of these things could have multiple functions. Some of them actually we're even very familiar with could have additional insights that we need to explore. So to sum this up, we think EV also, besides cGAS, besides toll-like receptors for the cGAS STING pathway, also is a very central and essential regulation mechanism to avoid, especially to avoid excessive immunological response. And this paper actually was actually one of the most viewed paper by Wiley in the first half of last year, just showing like the strong interest of EV, in general. Okay, cGAS STING, DNA, that's actually our machinery for to sense double-strand DNA. I used to work at this University of Colorado, Boulder. For you guys are familiar, like work in the RNA field. Sometimes we got the nickname, Boulder is the center of the RNA universe. So a lot of people actually around me actually like work on RNAs, and also we heard a lot of RNA story in the past couple of days already. So we had a joke in Boulder that talk about the God was seeing three scientists, and God was like, scientist A, so what do you work on? And he said, I work on protein. And God said, wow, the protein is so important. You got to sit on my left-hand side. And then the second scientist like, so what do you work on? Scientist said, I work on DNA. And God said, oh, DNA is even more important. You got to sit on my right-hand side. And then the third one, so what do you work on? And the scientist said, I work on RNA. And God said, okay, you take my seat. RNA is so important, apparently. And I still feel that we still actually need to learn a lot more things about EV-associated RNAs. So back in 2020, Dr. Catherine Xiao in the audience actually worked with Professor Ken Whitworth's lab, put up a perspective in JAV also, actually looking into speculating rather back then, I would say, different types of RNA shows in the EV-mediated cell-cell communications. And they could have actually many impact in different disease areas, such as aging, for example. Catherine showed that microRNA, mere 30B5P, actually was play important roles in regulating the cell senescence by actually regulating the third family protein's function. And recently, we moved also to the double-stranded RNA. So this actually is, I think, is a very exciting field. We do know, for example, the Ming lab at University of Pennsylvania showed EV-RNA effect innate immunity, and also from our lovely co-chair, Professor Ming Le's lab, Ming actually demonstrated very elegantly that RBC-EV carries double-stranded RNA, and we also talked about yesterday, so it's a very tiny microRNA could actually render a lot of potential in therapeutic development also. So Gaoge Sun actually also sitting in the audience. By the way, Gaoge also presented a poster, number 155. I would encourage you to see this excellent young guy and talk about his work also. He was asking, besides red blood cells, how about platelets? For EV, I guess you can't really get around with platelets, right? In healthy people, in our body fluid, in the blood, a major source of the EV from the platelets. Even in the field of hematology, people still actually like to prefer using the word microparticles, indicating those platelets-derived particles that actually regulate blood clotting process and things like that. So in cancer patients, if you're aware, actually one of the leading lethal cause is blood clotting. So they just actually tend to clot more. And we're actually asking the question really what EV shows in the tumor cells communication with platelets. Platelets is an interesting cell because they don't have nuclei. They can't actually produce proteins themselves. So when Gauger looked into different patients' sample, especially by different RNA, he actually found it interesting that the rpMRA, which is the component for the ribosome. Ribosome was made of different protein and RNA molecules, and for rpMRA, actually was, we still know so little about them. It seems in different cancer patients, the rpMRA underwent substantial decay. And he was looking at different, actually the rpMRA sequence in the platelets, and they all show actually significant decrease. So Gauger was trying to collect some of these cancer cell-derived EVs, and actually to see their impact on the microkaryocytes, because again, the platelets do not have the machinery to produce the proteins, and they were produced by the other cells. And seems as a consequence of that, this EV's treatment would cause platelet activation. We thought platelets, actually the resting state is actually rather this regular shape, and upon activation, they would actually underwent pretty dramatic shape change and triggers the blood clotting process. So we found this actually is really quite interesting. Again, we already talked about the cGAS-STING pathway, right, as a double-stranded DNA sensing machinery. Gauger was actually asking question, what about double-stranded RNA? It seems actually in the presence of ATP and also the OAS, that is in parallel to the cGAS-STING pathway for DNA enzyme that also synthesize this RNA cell that would actually triggering on the RNA decay process. So we found maybe it seems like in the presence of the double-strand RNA, the tumor cells use that to actually figure again to tell actually there's something changed, and then I want to actually as a consequence for the immunological self-protection machinery would actually trigger the decay of mRNA. So we found this actually indeed really quite interesting. And the tumor EVs seems, we do know, for people who work on RNA, you would know this is much more unstable than DNA. During the COVID period, when we do the environment test, DNA, RNA usually just was gone after several weeks. DNA could last sometimes for a year or so, and this really caused a lot of pain, especially when we did a lot of this nucleic acid tests during the COVID. So what we found, EV actually protect this extracellular RNA, and of course the free RNA got actually degraded, and those EVs were indeed actually taken up by the cells. And we think this actually is interesting. Also, we coined a term called the TAVUP for this process. Not only because this actually shows a interesting double-strand RNA shipping mechanism, but also actually may actually a rather non-conventional biomarker strategy. Usually we saw the different level of elevation of the markers, but for this case, maybe the rpMRA's decay could be indication for the disease such as tumor. Then how about mRNA's transportation? Another student coming to this SF 2024, Kaixiang Zhang, also was asking the question, really what the machinery help to actually pack, sort, and secrete those messenger RNA, right? RNA has all of different types. So there actually were interesting previous studies previously, but the specific machinery helped to pack the mRNAs for the EV secretion was still actually need a lot of elucidation. So Kaixiang, actually after screening, he identified a RA binding protein, or RBP, called NPM1, seems relevant. So in this figure, you can see that NPM1 not only co-localized with RNA molecules and also particle markers, it seems actually they even wrap this up. If you knock down NPM1, the mRNA, the EV-dependent mRNA transportation basically got blocked. So we found this actually is interesting. This actually could actually render some likely first insight of this sequence-specific RNA transportation shipping machinery. Again, by combination of bioinformatics and biochemical method, Kaixiang actually identified those candidate sequence. Actually, they're short. This actually, again, I think it's exciting also for potential technology translation, because when we attach those sequences to different mRNA, they could actually ship mRNA, push mRNA into the EVs. Could be a interesting strategy to ship those important nucleic acid molecules. So why did NPM1 do this? So we think it seems like those mRNA sequence was often found in different oncogenes, basically in the pathogenesis of cancers. So when we look into those sequences, seems they actually especially in the different carcinoma, for example, they were found. Maybe that's a way for cancer cells urgent needs to talk with the environment and shipping the messenger RNA out. So we showed this mRNA indeed actually get into the EV, not actually laying on the surface. They're not, I guess another was a term yesterday people talk about surface thing, but actually they were really inserted into the EV. And also this oncogeneous mRNA could be taken up by the cells. So we also showed this actually is relevant in clinical samples. Kaixiang collected samples from patients with different cancers at different stage. It showed actually compared to the normal EVs, the cancer patients really use this EVs, NPM1 dependent pathway to transport messenger RNA. So we speculate this could be a way for cancer really to stimulate the environment, actually affect for long distance communication and helping the cancer spreading basically also. So upon that, coming back to technology, we talk about the from bench to bedside. Back in 2009, we first worked on the curvature sensor. We try to actually build a peptide that sense particular curvature and lipid composition. Again, for curvatures, we also already heard some discussions at this meeting already. So you can define, for example, at this point, the curvature is that you fit a sphere, actually a small ball, snooker ball, for example. The smaller the ball is, the more curvature you have on the surface. You can just define this as the reciprocal of the radii of the ball, basically. So, for example, a point like this, a combination of C1 and C2 different curvature would give you a different shape. And we do know, apparently, in many biological processes, including EV uptake and secretion, membrane curvature play crucial roles. So, this is an AI time. I feel like at almost every single meeting, no matter what area you work on, people talk about computer science. So back then, a MD PhD student in my lab, Noah Castello-Weiss. Right now, he's practicing medicine in Palo Alto at Stanford, oncology. So back then, when he was a student in my lab, he used MD simulation, the molecular dynamics simulation, and while he was pursuing an MD degree, to show that with a highly curved membrane, just like EV, right? They're small, and they also have, because the small size, the lipids do not actually pack as well. They would actually give you the defects. So we're thinking, based on this characteristic, we can rationally design novel probe for those EVs, and really trying to grab the most disease-relevant EVs with small size with particular lipid component on the surface. So after I moved to Tsinghua, we also collaborated, have international collaboration with Jonel. Remember this guy who actually locates at Iloilo Island in Philippines? We try to actually use the first generation of the peptide probe for curvature sensor to actually detect this highly curved and lipid-specific curvature of different EVs. So this actually was, we have an international collaboration project, and this was when Jonel came to Tsinghua last year. And also actually I saw already, I have some the third generation, I mean the Philippine local students, they also got really strong interest in EV also. That's really encouraging. I feel like this, the academic line of the EV interest keep passing on. So for this, we just like to, because it was published, for the sake of time, I would just like to quickly mention, we have this fluorescently labeled peptide that specifically binds to a highly curved curvature of EV. For example, we're thinking just compared to the conventional ultracentrifugation method, it's much easier to use. So for example, this movie doesn't show, but this is a pathological rat model of blood. We can show in this complex environment, we add fluorescently labeled peptide, which actually show on this figure, only those small EV actually got labeled. So this could be a way for quick and economic alternative to the EV detection method. And we work with entrepreneurs also. Dr. Fred Lee, actually sitting in the audience, we co-founded Carrier Biomed based in Suzhou, which is an industrial city about 100 kilometers away from Shanghai. So we try to actually mobilize those curvature sensing peptide on the magnetic beads, and then it's actually pretty easy to use. Last year, we also came up with a machine that is fully automated. You can actually put the patient's sample in, and EV got out quickly. So even me, after 20 minutes training, I could actually use that myself. It's pretty easy to use, I can tell. And then we work with clinicians for clinical trials, with IIT. As I mentioned, Carrier Biomed is the sponsor of this meeting. So also, basically, also in collaboration with Leslie, Professor Andy Hill, and other clinician group in China, we work on different neurodegenerative disease and various cancer. So my time is running up. I'm not going to show you too much clinical data. We showed in different cancers, such as lung cancer, we can differentiate different stages, and also between patients from inflammation group and normal group. And for some other cancers, we actually can follow those cancer patients, not only the static snapshot rather. So indeed, we hope these things could actually provide useful information to the clinicians in their clinical practice. Finally, we disclosed the results for Alzheimer and Parkinson's disease molecular diagnosis. Actually, we test the blood EV in a group of more than 700 patients. We showed about 86% of accuracy, which is comparable to the gold standard currently in the market. So finally, I mentioned the people who actually did the work. Finally, thank you very much for your attention. Thank you.